Hello, hello everyone. In today's video lecture, we shall discuss regarding the formation of the nitrogenous waste products, that is ammonia, urea, and uric acid. So, in the previous class, we have discussed regarding the grouping of animals into ammonotelic, ureotelic, and uricotelic animals. So, based on the type of nitrogenous waste products that they are excreting. Now, as a part of the excretion physiology, today we shall be discussing regarding the formation of urea, ammonia, and uric acid. So, let us get started. Now, as we have discussed in the previous class, protein metabolism leads to formation of what is known as the nitrogenous waste products. Now, there are different types of nitrogenous waste products, five different types of nitrogenous waste products. So, majorly in them, we shall be considering regarding the ammonia, urea, and uric acid. So, how they are formed and how they are excreted out, that we shall discuss. Now, if we look at the entire animal kingdom, the formation of these nitrogenous waste products, so it is almost the same. Okay, so so because of that, if we discuss the mechanism, it is applicable to all different animals. Okay. Now let us come to the formation of first nitrogenous waste product, that is the ammonia. The formation of ammonia mainly takes place through two important steps. One is the transamination, another one is the deamination. Now, what is this transamination? Transamination is a chemical reaction where an amine group is removed from the amino acid. Okay, and it is transferred to a keto acid. Because of this transfer process, we term it as transamination reaction. Okay, and through this keto acid, the amine group will be transported to the liver, and in the liver, the keto acid undergoes oxidative deamination reaction, resulting in formation of ammonia. So this is the process through which the ammonia is formed in the body. Now, so what is deamination? As I have told in the previous slide, so here there is removal of amine group from the amino acid, and there is transfer to a specific keto acid in case of transamination. But in deamination, there is elimination of ammonia from the amino acid. Okay, so this amino acid will be converted into a specific keto acid, and during the course of reaction, since there is involvement of oxygen, this is also known as the oxidative deamination, and there is liberation of ammonia. Now, if you look at the protein metabolism, all these nitrogenous waste products are formed due to this protein metabolism. See, the proteins, they get broken down into amino acids, and amino acids in all tissues, almost all cells of the body, in the cytoplasm, they undergo transamination, resulting in formation of what is known as glutamate. And this glutamate or keto acid, it will enter into the liver. So in the liver, ammonia is formed. The ammonia formed in the liver, it will be detoxified in the liver through formation of urea, or it can be removed through urine in the kidneys. Okay, now the second step that we have seen in the formation of ammonium is the transamination reaction. Now, what is this transamination reaction? In transamination, from amine group from the amino acid is transferred to a keto acid. Okay, so and this reaction takes place in two steps. Number one, an amino acid is converted into alpha ketoglutarate acid, and this alpha ketoglutarate acid is converted into glutamate and pyruvate. And these two molecules, glutamate and pyruvate, that are formed during the transamination reaction, they pyruvate will enter into the Krebs cycle, and glutamate in the liver mitochondria, it undergoes degradation, forming ammonia. Okay. Now let us take some examples. One is the aspartate and another one is alanine example we shall take. So in presence of alpha ketoglutarate, these amino acids they get converted into glutamate. And the glutamate that is formed in presence of these two enzymes, one is SGOT and SGPT, very important enzymes. Both the enzymes have PLP as the coenzyme. In presence of these two enzymes and the PLP, these amino acids get converted into glutamate. Okay. Now, the expansion of these enzymes you can see in the note. Now, in the second step of the transamination, now what is happening? The glutamate that is formed, it happens inside the mitochondria in presence of the NAD plus and water molecule releases our important molecule that is ammonia. Okay, so here there is a release of ammonia in the second step of the transamination. Now, first step, what happened? There is a transfer of an amine group from amino acid to a keto acid, and in the second step, there is the keto acid that is formed, the glutamate that is formed in presence of NAD plus, it liberates ammonia. Okay, so this is the oxidative deamination step. Okay, so in almost all the tissues of the body, through transamination, amine group is transferred to a specific keto acid, and that keto acid comes to the liver. In the mitochondria of the liver, the specific keto acid in presence of NAD plus gets broken down and forms liberates ammonia. Okay, so this ammonium formed is very toxic. And what happens to this ammonia? So these are the steps that I have discussed, and you can see the final product of ammonia formation through transamination and deamination is the ammonia. Okay, so ammonia that is formed. So this is the reaction I was talking about. This amino acid, keto acid, and in presence of amino transferase and pyridoxal phosphate, there is formation of the glutamate. This glutamate enters into the mitochondria. In the mitochondria, the glutamate forms NH4 plus. Okay, so this step is known as oxidative deamination. Okay, so let us move on. Now, there is, apart from this deamination of amino acids, transamination and deamination of amino acids, there are also other methods where there is formation of ammonia. One is in the intestine where the uh, bacteria degrade urea into ammonia, that is one thing, and deamination of adenylate acid also results in formation of ammonia. And catabolism of purines also result in formation of ammonia. Now, what happens to the ammonia that is formed? Now, ammonia that is formed, first step, it may react with ketoglutarate, result in formation of the glutamate. Okay. Now, the glutamate formed can result in formation of what are known as the non-essential amino acids. That's the first fate of ammonia that is formed. It may be engaged in the formation of new amino acids. And also amination of glutamate forming glutamate. So remember, glutamate is a major storage form of ammonium, and also if it has to be transported, it will be transported in the form of glutamate. But most of the ammonia that is formed will be converted into urea. So this is a process of detoxification. So that is regarding ammonia formation, deamination, transamination, deamination occurring in the mitochondria, and transamination reaction occurring in the cytoplasm of almost all the cells, and formation of ammonia. So that ammonia formed, especially in higher animals which cannot afford to waste a lot of water excreting ammonia, this will be converted into urea. Now, urea formation, first of all, occurs in the liver. It occurs through a cycle known as the Krebs Anslet cycle. Now, the urea cycle occurs in different steps. Just a second. Different steps. In the first step, what happens? There's a carbon dioxide molecule which combines with ammonia. In fact, the carbon dioxide in the form of bicarbonate ion, HCO3 minus, carbon dioxide in the form of HCO3 minus forms an important molecule known as carbamyl phosphate. Okay, so in presence of the enzyme carbamyl phosphate synthase and with the consumption of two ATP molecules, there is formation of carbamyl phosphate in the first step of the urea cycle. Now, the next step, the carbamyl phosphate that is formed reacts with a special amino acid called as ornithine, which enters into the mitochondria from the cytoplasm, reacts with carbamyl phosphate, resulting in formation of citrulline. Now, the citrulline that is formed reacts with aspartate and results in formation of the organosuccinate. Okay, so this is another important molecule that is formed with the combination, with the combining of citrulline and the aspartate. Now, organosuccinate that is formed breaks down with the liberation of fumarate and in the course of liberation of fumarate from the arginosuccinate, there is formation of this important molecule that is the arginine. Arginine formed in presence of enzyme known as arginosuccinase. Now, this arginine form, it forms two important molecules. One is it results in formation of the ornithine, and while forming ornithine, there is formation of are important molecule that is the urea. Okay, so there's a last step of the urea cycle or the ornithine cycle or Krebs cycle where it is resulting in the formation of urea. Okay, now this ornithine that is formed in the cytoplasm again enters into the mitochondria. Okay, so ornithine enters into the mitochondria, again reacts with the carbamyl phosphate. A carbamyl phosphate comes into the cytoplasm, and in the cytoplasm it combines with fumarate, forming the arginosuccinate. Okay, so let us see the cycle. So these are the 5 steps under which the urea cycle takes place. See, as we have discussed in the previous points, ammonia reacts with the bicarbonate, resulting in formation of carbamyl phosphate. The carbamyl phosphate, in, uh, along with ornithine, carbamyl phosphate reacts with ornithine in presence of this enzyme known as ornithine transcarbamylase, forming uh, what is known as the citrulline. For formation of the citrulline, there is requirement of ornithine transcarbamylase enzyme. This citrulline that is formed along with the aspartate, both of them together, they form what is known as arginosuccinate. Now, this arginosuccinate formation takes place in the presence of this enzyme known as arginosuccinate synthase. Now, this arginosuccinate cleaves, liberating the fumarate, and while liberating the fumarate, it results in formation of arginine. The enzyme required for this is arginosuccinase. Now, arginine reforms the ornithine in the cytoplasm, which again enters into the mitochondria. And while doing so, in presence of organase, there is liberation of urea. Okay, so this is how urea is formed. And this reaction, where ornithine reacts with carbamyl phosphate, all these reactions, this reaction occurs in the mitochondria. Okay, and these reactions that we are referring to, they occur in cytoplasm. Okay, and in fact, when there is formation of ornithine in the cytoplasm, it enters into the mitochondria to react with the carbamyl phosphate. Just remember that. Okay, so let us move on. Now, the third nitrogenous waste product that is formed is the uric acid. Now, uric acid is mainly formed through what is known as protein degradation, or purine nucleotide degradation. There are two important purines. One is adenine and guanine. Now, degradation or the metabolism of these purines result in formation of what is known as uric acid. Now, let us see the steps. So, look at here. There are three important metabolic products in the body. One is AMP, another one is GMP, another one is IMP. What is AMP? Adenosine monophosphate. What is GMP? Guanosine monophosphate. And what is IMP? Inosine monophosphate. These three molecules are the molecules which are carrying the purines along with them. So adenosine monophosphate, inosine monophosphate, and guanosine monophosphate. These three molecules, uh, they undergo degradation, resulting in formation of the uric acid. Let us see how it happens. The adenosine monophosphate gets converted into what is known as inosine in presence of this important enzyme known as adenosine deaminase. So this adenosine deaminase converts adenosine into inosine. Inosine in presence of nucleoside phosphorylase forms hypoxanthine. So in presence of nucleoside phosphorylase, it forms hypoxanthine. So it is just 1x. There's a correction. Um, Hypoxanthine can be formed from the adenosine. Now, adenosine and inosine results in formation of hypoxanthine, and hypoxanthine forms xanthine, and xanthine forms uric acid. This is one. And another molecule that is present, guanosine monophosphate, forms guanosine, and guanosine results in formation of the xanthine. Okay, so formation of guanosine into xanthine, and again it gets converted into uric acid. So remember this important enzyme. So these are the very important enzymes that results in formation of the xanthine and hypoxanthine molecules. So through this purine degradation reactions, there is formation of the uric acid. So three important nitrogenous waste products are formed. One is the uh, for protein degradation results in formation of ammonia. Now, ammonia form gets converted into urea. Uh, in some animals, we get excreted in the form of ammonia only. So, aquatic animals excrete ammonia. Ammonia is formed through two important steps. One is deamination, another one is transamination that we have discussed. And ammonia forms urea in higher vertebrate animals, and urea formation takes place through ornithine cycle or the Krebs-Ansett cycle. Now, the uric acid formation takes place through what is known as purine degradation. So there are two purines. One is the adenine, another one is the guanine. So adenine and guanine undergo degradation, resulting in formation of what is known as the uric acid. Okay, so this uric acid is comparatively least toxic compared to urea, and urea is less toxic compared to ammonia. Okay, so that is regarding formation of urea, ammonia, and uric acid. Thank you all.